Susan, and welcome everybody. Mark and I are really excited to tell you about the GeneSYS project and where we are in that. And I think, it's been some exciting progress so far. So glad to share that with you. Let me advance. So just a little bit about the background and kind of the scientific rationale for why we are approaching this the way we are. Um, in everyone who's kind of walking around who does not have Schaaf-Yang syndrome, they have a functional copy of the MAGEL2 gene. And every person has one copy that was inherited from their father that is producing protein and doing the things that MAGEL2 should do. However, in our loved ones with Schaaf-Yang syndrome, they have a mutation in that gene, and that makes a protein that is not full length. So all of the mutations that have been identified to date cause these what we call truncated proteins. So it essentially is making a shorter protein. And the part of the protein that is missing is really important in directing that protein to a certain place in the cell, and also important for interacting with other proteins in the cell. And what happens when we get this truncated protein is it goes to the wrong place in the cell. So if we look on the right-hand side of this slide, this is a single cell. The blue part is the nucleus, that's where the DNA sits. And the other part is what we call the cytoplasm. That's essentially the body of the cell. And in this particular cell, the MAGEL2 protein is it is tagged with a green fluorescent tag. So the MAGEL2 protein, you can see it all throughout the body of the cell or the cytoplasm of the cell. This is a normal MAGEL2 protein. So if you were to take a cell from anyone who does not have Schaaf-Yang syndrome, that's where the MAGEL2 would go to do its normal function. In a Schaaf-Yang syndrome individual, the MAGEL2, as we said, is truncated, and rather than staying nicely in the cytoplasm, a lot of it moves and gets stuck in the nucleus of the cell. Now, there's always a little bit of MAGEL2 in the nucleus of the cell, but it's very different in Schaaf-Yang syndrome, where it seems to be accumulating in the nucleus. And we believe that is what is causing the cell not to function in its typical way in Schaaf-Yang syndrome. So then what can we do about that? And another piece of information that has helped form this project is that there are very rare cases of individuals who are just completely missing the MAGEL2 gene. So we know in Prader-Willi syndrome, individuals are missing the MAGEL2 gene, but they're missing several other genes as well, and they have some characteristics that are shared with Schaaf-Yang syndrome, but also some that are not. But in these two very rare individuals who have been identified to date, they're missing just the MAGEL2 gene. And actually, both of them don't seem to have many symptoms at all. And so this leads us to the hypothesis that maybe it's the MAGEL2 truncated protein that's getting gummed up in the nucleus of the cell that is causing the issues of Schaaf-Yang syndrome. And if we were able to just delete or remove or get rid of that protein, we might end up with a situation that is actually better for the individual with PWS. So that idea of getting rid of a mutated protein is a concept that has been seen in other rare diseases as well. Probably the best known is Huntington's disease. So that is also where a mutation causes the protein to kind of be in the wrong place and do the wrong thing. And if you get rid of it, you actually have a better clinical situation than if it's present and mutated. So this has led us to think about, okay, so what can we do to eliminate that truncated, mutated MAGEL2 protein in individuals with Schaaf-Yang syndrome? And one of the more common ways to go about that is this essentially a drug that's called antisense oligonucleotides. And this is a well-established therapeutic pathway. ASOs, which is the abbreviation for antisense oligonucleotides, have been used in a number of different diseases and disorders in cancer to get rid of proteins that are contributing to the cancerous state in other genetic disorders as well. So the FDA is pretty familiar with these ASOs and kind of the studies that need to be done to take it into clinical trials. The ASOs themselves are short pieces of DNA and RNA, and they match exactly the gene that you're trying to eliminate. So we have this sequence-specific binding of the ASO to its target sequence, and what that does is it blocks the protein from being made. And it causes the RNA to be degraded. Um, so we want to apply this ASO technology to Schaaf-Yang syndrome to eliminate the mutated or truncated MAGE-L2 protein. So there's a pretty well-known process that one needs to take to go about that, and we have started down that pathway with this GeneSYS project. So to date, we've really focused on identifying where exactly in the MAGE-L2 gene we want our oligo to bind, because depending on where it binds, it's going to have better or worse efficiency at getting rid of the encoded protein. So I will pause there, see if there's any questions about the rationale, and then we'll let Mark kind of walk us through where we've gone so far. So does anybody have any questions about the general approach or, why we're taking this approach for Schaaf-Yang syndrome treatment?
And if there are questions, you can come off mute and ask them directly, or you're welcome to throw them into the chat and we'll monitor the chat for questions as well.
Okay, if there's no questions right now, then I'll turn it over to Mark to tell you a little bit more about the strategy that we're using to develop these ASOs.
Thanks, Teresa. Yeah, I'm really happy to talk about this today. And as Teresa said a couple of times, ASOs are pretty well established, and the development pathway, at least the first couple of steps, have become very standardized to the point where contract research organizations like the one we use, Charles River Laboratories, they already have these package deals that are set up to move carefully through the steps that will ensure that if you find an ASO, it will be well tested and it looks like we have a question. Oh, we can catch up with how you can help at the end. So in this initial step, which is to identify the sequence of the ASO, ensure that it's getting rid of the MAGE-L2 in the cell, and then checking the safety and make sure it's not interacting with other genes. The project so far has been set up with these 5 phases. Phase 1, designing the ASOs. So as Teresa said, the ASO is a short piece of nucleic acid, and it's less than 1% of the size of the MAGE-L2 gene. So there are a lot of different spots in the MAGE-L2 gene that you can design the ASO to attach to. And there's no way to predict where the binding will be most effective at stopping production of the MAGE-L2 proteins. So in this first phase, we design all possible ASOs that would bind to the MAGE-L2 gene, and then computationally identify the top 100 for the binding chemistry and make sure that they are not predicted to bind to any other genes in the genome. And once you have those 100 designed, you can synthesize them, and then they'll be ready for testing. Phase 2 is to ensure that the laboratory testing the ASOs will actually be able to detect if they work or not. And so the assay being developed is to is to measure the amount of MAGEL2 being produced in a cell. And they have to make sure that they can measure it well enough that if the ASO reduces the amount of MAGEL2, they'll be able to detect that change. So you need a sensitive ability to measure the MAGEL2. And if you can't do that, there's really no moving forward. So that's something that you have to do early in the process. That's why it's set up this way. Phase 3 combines the results of phase 1 and phase 2. You take all of the ASOs that have been designed that bind to different parts of the MAGEL2 gene, and then you use that assay to determine to what degree each of those ASOs can reduce the expression of MAGEL2. Since you're screening 100 different ASOs at that point, we just use them at one concentration each, and then we can determine like how much MAGEL2 is reduced. The top performing ones move to phase 4, which we call potency. And you can see in the smaller print there, optimizing dosage. So the top ASOs will be tested at different concentrations. Usually you expect that if you take more of a drug, then the effect will be stronger. And so that's what we're looking for is to see that increasing the concentration of the ASO increases the knockdown of MAGEL2, and that shows that it's working the way it's supposed to. It also helps us to understand what dose may be required to get optimal knockdown. You don't want to give too much if it's not really increasing the level of knockdown. And then those have all been completed successfully, and we're kind of in the middle of phase 5 now. We're very close to the end of phase 5, just analyzing the data, and that is to ensure that no other genes are being knocked down by this ASO. And I mentioned in phase 1 that computationally you can look at the sequence, you can say this is probably not going to bind to any other transcript or gene in the genome, but you want to prove that. So what phase 5 does is it looks at all of the transcripts in the cell and ensures that only MAGEL2 is being affected, or potentially things, I guess, that might be downstream of knocking down MAGEL2. But this helps us understand if the cells are still healthy after treatment with the ASO. And so for phase 5, you take the best performing ASOs from phase 4. So Teresa, if you'll advance the slide, I'll talk a little bit about how these went. So this project, I skipped over the last slide. This process was quoted to us at about $500,000, and thank you for your donations to and fundraising efforts to help us get there and buy this. And it started last March, and so I like to highlight how efficient this process is, having started in March, and then in less than a year, we will have completed all of those steps. So phase 1, the designing, and phase 2, the development of the assay, were completed within 2 months of starting the project. It took a couple of more months to get the screening set up, and because after you grow all 100 cultures of cells and put the ASOs on there, you have to then perform the assay on all of those cultures and quantify how much MAGEL2 is there, how much has been removed as a result of treatment with the ASO. And so you get the kind of data like the upper chart there, where each bar shows how much MAGEL2 the cell is expressing. And so the colorful ones on the left are controls. And then the ones on the right, fortunately, are all a little bit lower than the controls. And the lower the bar, the more MAGEL2 is removed. And you can see some ASOs like 65, 67, they didn't reduce the level of MAGEL2 at all, but 71 and 72 did pretty good. And so looking at these all at kind of a standard concentration, these were human cells, Carmine. I saw that pop up. Yeah, so we have focused on the human sequence, and all of these ASOs are designed to bind to the human transcript, and so we're using human neurons for this. And it has to be neurons because they have the highest level of expression of MAGEL2, and so we need, of course, to be able to detect MAGEL2. It has to be there for us to get rid of it. So I put a few numbers in that bullet there. The best ASO that we had reduced the expression level by 80%. We had 2 that were more than 70%, 5 that were over 60%, and 9 over 50%. And if the reduction is less than 50%, then there's not much point in further work with those specific sequences, although you can optimize them, look at places in the gene close to that. So we took those top 9 that were over 50, and since there was enough space to do 15 in the dose ranging phase 4, we actually synthesized 6. We designed 6 new ones close to that 71 and 72 that were the best knockdown, thinking that this is a hot spot in the gene. And ASOs that bind here will be more effective. So we designed 6 new ones. And so we had 15 that went into phase 4, the dose dependency. And that's kind of some of those data are in that second figure, the lower one. And from left to right, you have an increasing dose of the drug. And from bottom to top, it shows how much MAGEL2 is being reduced. And so the pink one is a kind of you can see very clearly that the more drug, the more MAGEL2 is eliminated. And so these are behaving the way they're supposed to be behaving. And so that gives us confidence in moving forward. Then the top 5 cultures from that experiment, and that meaning top 5 being the most MAGEL2 knockdown. So it's a combination of the ASO sequence and the concentration that seems to be best. Those were sent for the sequencing of all cellular RNA to make sure that it's not affecting all of the genes. And so that sequencing has been done. The data are being analyzed, and we were told that the data should be ready by the end of this week. So we have a meeting coming up, and we'll talk about that. And that will complete phase 5. And when that is done, we'll have completed kind of this our initial set of goals, which is to find an ASO that effectively removes MAGEL2 and doesn't significantly alter expression of any unrelated genes. So maybe looks like there's a question. Karen, you want to unmute?
Yes. Hi. How do you geneticists did it when they were recently born or whenever?
So these are actually model cells from healthy patients, neurotypical patients, and they're the reason that we needed to start with healthy cells here is that we want a maximum level of MAGEL2 so that we could see that we're removing it. We're not yet at a point where we're removing it from any Schaaf-Yang cells.
Oh, okay. So no Schaaf-Yang children have been tested yet.
No, in fact, no children at all. It's just cells in a dish at this point. And then
when
the
you
next
do
steps,
test them, is it blood work, or is it urine, or is a cell from a cheek, or what is it that you'll be testing?
Well, when we eventually test in Schaaf-Yang model cells, we haven't selected the cell line yet, but they could be engineered from a healthy cell with Schaaf-Yang mutations introduced by CRISPR. The benefit of doing that is then you know that the genes, the genotype of those cells is identical in both control cells and the experimental cells. We would also want to check, as you know, there are many different mutations that can cause Schaaf-Yang syndrome. So we will want to ultimately, and this is a couple steps away, but we'll want to test cells representing different mutations because some patients have a very short piece of MAGEL2 remaining, some have a longer comparatively piece of that. So these cells that can be made from patient samples, we can make them from the blood that Combined Brain collects for us. They can be made from patient skin or cheek swab, like you mentioned.
Okay.
I don't think that spit is a very effective source, but the key thing here is that, like I said, it has to be a neuron because only nerve cells express MAGEL2. So if I got a skin cell from you, I would have to then turn it into a stem cell in the lab so that I could turn that stem cell into a neuron. We can't go
Wow.
sampling neurons. It's
Okay.
quite dangerous to sample neurons from living people.
And then MAGEL2 came from dad, you said, Teresa?
Yeah, so typically in typical people, all of us, right, the MAGEL2 that was inherited on our dad's chromosome is the one that's active.
I
So
get it. Okay.
both PWS and Schaaf-Yang are unusual in that it matters if it was the chromosome that it was inherited from your dad or your mom. And most
Okay.
genetic disorders, it doesn't matter, but this one is unusual in that.
Okay. So de novo doesn't mean not from dad or not from mom.
It just means that in dad, the chromosome was fine, but as it was passed to the child, there was a new mutation that was introduced.
Gotcha.
It
Oh,
happens
I understand.
kind of
Thank
all
you.
the time, if you look at parents and their children, there's all kinds of new mutations, parent to child. It just happens. Sometimes those happen in areas of the DNA that don't matter, and sometimes they happen in a gene like MAGEL2, and then you get a genetic disorder because of that.
Well,
So,
thank you both.
yeah.
But yeah, and I have a question for Mark. Mark, you showed the slide that had the five phases of the work you've done so far. What does that next slide look like with the remaining phases? And is it, another five, or is it, 250 phases?
So I think that's what's on this slide, and maybe Mark, we can there's just I think this one and one other, and then maybe we can. I see there's lots of questions that are popping up, so maybe we can just get through this part, and then we'll see what questions remain.
Thank you.
I do want to do one question that I see, which is one of my favorite questions from Mark Polankovsky, if I'm saying that correctly. And Mark asks that since we're using healthy neurotypical cells with full-length MAGEL2, will the drug still work on truncated MAGEL2? And this is actually, it's a really good question. And I thought that myself at the beginning, but then it occurred to me that the ASO is actually affecting the production of MAGEL2 at a point where you still have the full sequence being transcribed from the gene. And so I would have to go back and kind of teach how RNA is made from DNA and protein is made from RNA, but you still have the full length RNA that has that mutation in it. So these ASOs, even if it binds to a point past the mutation, it'll still remove that RNA and then the downstream proteins. So these would be effective for any Schaaf-Yang patient with any mutation.
And actually, that's one of the reasons that we are going this route. There is CRISPR technology which can do each mutation, but you would have to have a different CRISPR for each mutation. The ASO technology should be applicable to all Schaaf-Yang mutations, and that's one of the reasons that we sort of chose to go this route first, although we are still paying attention to the CRISPR technology and seeing how we might apply that.
Right. So how many steps are left? And I'm not going to talk about testing in humans because you probably know how clinical trials go through phase 1, 2, 3. We can cross that bridge when we come to it, but we can talk about it more later if you want to. But so far, what we have shown is that we can reduce expression of MAGEL2, but it's very important to show that actually improves the health of the cells, like our initial hypothesis based on those patients that didn't express MAGEL2 but were largely asymptomatic. And we haven't proved that. We haven't actually shown yet that the cells become any better. So Number one, we're patenting these sequences. And that is because if we ever want to partner with a commercial entity to further develop this stuff and get a drug out to patients, they'll need IP protection. They need to make sure that they're the only ones able to sell this and make a profit from doing it. So we'll patent this, we'll hold the patent and license it, or make deals with people to get FPWR some money eventually, and then continue our work. But in order to prove that hypothesis that that blocking MAGEL2 expression improves the symptoms, we are still designing the experiments for that. And so we don't have a quote or a timeline yet, but this is how it's going to go. We like to start in cells because because it's a more controlled and simpler system to look at the measurements. And so what we really need to do is find a measurement that can be done on cells that can tell you the difference between a cell with a truncated Schaaf-Yang form of MAGEL2, a cell with no MAGEL2, and then the control cell with the full-length MAGEL2. And unfortunately, cells don't always act exactly like humans do. And so those patients that have no MAGEL2 and are asymptomatic, the situation in cells, at least the published data so far, has seen similar effects in MAGEL2 null cells, cells with no MAGEL2 and cells with a truncated MAGEL2. Although new data, kind of not yet published, is leading us in directions that that we think we'll be able to actually tell the difference here. And so we'll be able to measure that, find something measurable in the cells that'll show us that the Schaaf-Yang cells are improving in some unhealthy activities and becoming more like a healthy cell. And so, yeah, that challenge is it's not a brain. We don't really understand why brains don't do exactly things based on the way cells behave. But we're feeling good about the choice of assays at this point. And so we're just going to work with our CRO to make sure that they can do the assays, and similar to detecting MAGEL2, like I talked about in phase 2 of the earlier work, they're going to optimize these assays so that they can measure a change in the cells when they're treated with the ASOs. We also would want to then do this in an animal model. We have well-characterized rodent models, and rodents, there are more things you can measure than in a single neuron. The Schaaf-Yang model rats have very unique feeding behaviors, social behaviors, motor behaviors. They walk differently, and performance in cognitive assessments. And so these are all things that cells don't do on their own. And so when we treat these mice with the mice or rats with the ASOs, we'll be able to make more measurements to look at a difference between essentially, you're turning a rat with a truncated MAGEL2 into a rat with no MAGEL2. And so those are the things you have to be able to distinguish. The problem with going to a rodent is the gene sequence, the sequence of MAGEL2 is so different that we would have to redesign the ASOs and find sequences that are specific to the rat. So that's not necessarily something we want to do as an initial next step. We're going to continue working in cells first, but then in the future, when it's justified, because the cells seem to be improving, then we can move forward to the rodents. And we thought it would be good to provide a brief look at some of the other projects that we're sponsoring, because they are all kind of strategically aligned to meet our goals in advancing this program. We knew these questions were coming, and so we've had people working on these for some time. Magdalena Lausch in Heidelberg is using has very advanced new ways of looking at these cells to try to distinguish the Schaaf-Yang mutation cells lacking MAGEL2, PWS cells, neurotypical cells. And this is actually leading us towards some of the assays that we think are going to be used to test the ASOs. She's detecting things at a level of sensitivity that we haven't seen before. So watch for her publications, and we'll blog about them when they come out. Kelly Rabianet in Barcelona. She's focused on the activity of rodent MAGEL2. Of course, I talked about we have rodent models, they have unique behaviors. People always ask, are you sure that in the rodent, the protein is doing exactly what the protein does in a human? And so we're going to de-risk that rodent model by tackling these questions one at a time. Does the rodent one do the same thing as the human? So she has shown the rodent one accumulates in the nucleus, just like the human one that Teresa showed you on the first slide. And that was a big unanswered question. No one had ever looked at the rodent MAGEL2 subcellular localization. And because we recognized that that was going to be a question, we worked with Kelly to set that up. Rachel Gilmore, you may have heard about, also in Heidelberg, working with Christian Schaaf, who I'm sure you've heard about. So she's doing a postdoctoral fellowship. And so this is a period in training. After you earn your PhD, you take a couple of years, focus on a project really deeply. And she has chosen to spend this time of her life looking at MAGEL2. And she has... among several projects that she's got going on, some focused on assay development for MAGEL2 activity, some on genetics and heritability of the mutations. She's also kind of following a parallel path to us, but developing ASOs for the rat. And so she is starting with the rat sequence, designing ASOs based on the rat sequence, and then she'll be using rat cells, looking for knockdown in rat cells, and then eventually treating the rats and looking at the behavioral phenotypes there. And she was one of the authors on this paper that came out just last year, which describes the rat in a very high level of detail and is going to be a model for all future rat studies, because this paper that I'm showing here kind of defines all of these measurements of the rat. And so we'll use that kind of as a model to go back. Interesting question from Chris Cooper, why not use a humanized mouse? And at this point, the reason would seem to be that we don't need it yet. We haven't found any evidence that we need to make a humanized rodent. Of course, we know we can, but it takes time and money to do that. And fortunately, because Dr. Rabianet's work has not found any significant differences in activity between the human and rodent MAGEL2, we don't think that that's going to be necessary, but it remains an option if we have to. Yeah, then knockdown, we could then test the knockdown because it would be the human sequence. So that would be one thing.
So maybe we can just end quickly with
Yeah.
some ways that you can help support this research. And then I know there are a lot of questions, so we'll save time for that. So we'll just mention again that we have the Global Schaaf-Yang Registry. I see there's a question in the chat about a little boy. Yeah, a little boy who at 3 years and 1 month had been talking and is now suddenly stopping. And I think the registry is a great place to be capturing some of the developmental We're going to be adding some new surveys to the registry so we can catch more of that information. But there's not many kids and adults with Schaaf-Yang syndrome, so understanding the broadness of the different symptoms and how development goes, I think is going to be really informative. And the registry is one place that if everybody contributes data to that, we can get a much fuller picture of, how Schaaf-Yang how individuals with Schaaf-Yang develop, the challenges that they face, and the broadness of that spectrum. So please do consider joining the registry. If you have any issues joining the registry or answering the questions, you can contact me. I will give you to Jessica Bahanowicz on our team who can walk you through step by step on that. So that's a huge, important resource for the Schaaf-Yang community. We also hope that you continue fundraising for Schaaf-Yang research. There's a lot to be done, as you can see. We're trying to be as efficient with the funds as we can, but these development of therapeutics that go into humans is expensive. And finally, we do have a biorepository through Combined Brain. Please look on our website for those of you who are in the US to see where you might be able to donate, because there's different donation spots at different times and places across the country. So please keep an eye on that. And if there is a opportunity near you, please go by and consider donating samples for that.